All right, so in this one, I'm going to go over question number 2 from 2017. This one is going to be pretty similar to what I feel like you guys would get on this year's exam. It's really analytical. A lot of it is just looking at data, scientific method. It looks intense. When you break it down, it's really not too bad. So for number 2, it talks about fireflies frequently occur in some ecosystems. They can destroy all above ground vegetation. Many species of plants in these ecosystems respond to compounds like smoke that regulate seed germination after a major fire. So we got carrikins, which are abbreviating KAR, and trimethylbutanolides, which are going to be abbreviated TMB, are water-soluble compounds found in smoke that are deposited in the soil as a result of a fire. So it's telling you that the TMB are water-soluble compounds. And why does that matter? Well, it's not asking anything about it quite yet, but if you forgot what water soluble means, water soluble, if you forgot, it means it's polar. So water soluble, we're going to call as being polar. Okay, that means it is going to eventually dissolve in water. So understanding that makes this quite a bit easier. So it says CAR and TMB bind to receptor proteins in a seed. In a study on the effects of smoke on seeds, researchers recorded timing and percent of seed germination in the presence of various combinations of CAR and TMB. The results are shown below. So this one is showing you, it looks like we got 4 groups. We have CAR, we got a control, we got CAR with TMB, and then just TMB alone. So they're, over time, they were measuring how much seeds actually germinated. And just to kind of draw this picture out, I like to have this to make some sense here, where if this is the seed, these receptor proteins are definitely not drawn to scale, but it's saying that TMB will bind to this receptor protein, and then CAR binds to this receptor protein, and the claim is they both have an impact on how they germinate. The control, just understand this, that they still have those receptor proteins, but they're just not adding any one of these. So in the second investigation to the effect of water on seed germination after a fire, researchers treated seed with CAR or TMB. The treated seeds were then divided into 2 treatment groups. One received water rinse and the other received no water rinse. The seeds were then incubated along with the control seeds. The results are shown. So let's look at this first group. We'll kind of go across and make sure you guys feel comfortable how to read it. So if I had group 1, it says that they did not, the minus means they didn't receive it, so they didn't add CAR or TMB, they didn't add water, and they have a result of the control. For group 2, they added CAR only, no TMB, no water, and the results were different from the control. With group 3, they didn't add CAR, but they only added TMB. Their results were different. Group 4, they didn't add any, but they did add water, but they still received the same result as the control. 5, they added CAR, no TMB. They did add water, and the results were still different than the control. In group 6, they didn't add CAR, but they did add TMB, but they added water, and the results were then the same as the control. So right away, this one should kind of be jumping out at you, and we'll talk about that here shortly. So let's look at the question on what you actually needed to answer to earn some points on this. So A, it says the researchers made the following claim about the effect of CAR and the effect of TMB on seed germination relative to the controlled treatment. So CAR alone affects timing, CAR alone affects the percentage of germination, TMB affects timing, and TMB alone affects percentage. So it even tells you to justify these, or you got to provide support from figure 1 for each of these claims. So basically you want to just understand and how do you support these claims. So they say that CAR alone affects the timing of seed germination. So in all honesty, this 4 points from part A is really just about reading this graph. So CAR alone affects the timing. Let's look at this one first, the timing of seed germination. So for time, let's look at this. We're looking at CAR and we're comparing it to the control. So actually, let me jump back to this. Regardless of all these, you have to essentially always compare to the control group. If you get a question like that, then it makes you justify using a figure. Most of the time, they want you to make sure you're comparing it to the control, granted if they give you one. So let's look at CAR in relation to the control. So that means there's none of those signals added. So in this part A, we're looking at time. So we're going to be looking down here. It looks like right here with CAR alone, it's going to actually germinate at 12 hours. whereas with the control, it doesn't look like it starts to germinate to closer to 24. So let's answer this one. CAR alone affects timing. How do you explain it? I would say that CAR germinates at 12 hours, where the control starts at 24 hours. So that's how you get that one. Pretty simple. For the next one, how does CAR alone affect the percentage? Again, you're comparing it back to the control, but in this case, now we're actually looking at the seeds germinating. So it looks like the control group received almost 80%. It looks like with CAR, you do have some, looks like over 90%, almost 95. So you're just basically going to state how does that compare to the control. CAR has a 95 or 90. I bet you they would give you a range here. CAR has a 95% germination rate, whereas control only has, control is going to have about, what was that value? It looks to be around, I'll just say 78%. So it definitely increases it. So CAR increases the timing that they germinate, but CAR also increases the germination rate. So that's going to be those simple 2 points on that one. So then we got to look at it. How does TMB affect the timing of seed germination? So we're comparing it now to the control. So we're going to be looking at TMB compared to the control group, and we're looking at time. So TMB and versus the control, it looks like right around maybe 20 hours, the control starts to germinate, where TMB lags behind the control. So TMB alone affects the timing of seed germination. So I would say that TMB germinates after the control, let's say at 24 hours. So that's how you would explain that one. And then the last one is going to be pretty similar. How does TMB alone affect the percentage of seeds that germinate? If you look at it just in terms of the percentage, TMB has a very low germination percentage compared to the control. So you would have to just clarify and say that TMB is actually going to decrease the germination rate. So TMB is actually going to decrease the germination. Over control. So basically, it's a lot of this is just analyzing the graph itself. So yeah, let's kind of make sure that we feel good on those. And this was actually 4 points, and again, a lot of that was just based off of reading the graph. So I'm going to keep that. I want to say I had the rubric pulled up here. Yeah. So let's look at here. Basically, yeah, the support is what we just went over. You're just analyzing the graph. So hopefully that one wasn't anything that is too difficult by any means. Now for part B is a lot going on. It's not any more difficult, but it says make a claim about the effect of rinsing on binding of CAR to the receptor in the seed, and about the effect of rinsing on the binding of TMB to a receptor in the seed. Identify the appropriate treatment groups, and results from the table that, when compared to the controls, provide support for each claim. So this one is actually worth 4 points. A lot of this is just summarizing this table. So you're going to get a lot of your points just based off of analyzing this data. So let's look here. We're now going to be looking at this experiment where this is their control group. Okay. Didn't add anything. There's your control result. When you look at group number 2, you need to understand that when you added CAR, you didn't rinse water. It's different than the control. Adding TMB, it's also different than the control. But then now when you added in water as the variable, because all 3 of these groups 4, 5, and 6, you added water, it makes sense that these 2 are the same because you're not adding any of the hormones. But the one new one that is important to notice is this one, is it's the same as the control. So when you compare this, let me use a different pen color to help emphasize this. So let's look at this one. When you have TMB, right? When you added in TMB without water, it was different than the control. But when you compare added in TMB and you actually mixed in water, you're getting some different results. So that tells you something about TMB, okay? So let's jump back here. Basically, when you saw this adding in TMB, adding in CAR, there was a key thing that it told you in the problem to help this problem become a lot more manageable. So, and that was talking about water soluble, okay? It tells you that TMB are water soluble compounds found in smoke. So first, let's look at this. TMB, if you see it, the key thing is that it actually is going to decrease germination. Okay. So just by looking at the data, having TMB present is important because it actually decreases germination. So basically what happens is if you had a water solution that was added, right? So if you added in water to this, okay, it basically, you have to answer this question and assume that, okay, well, no TMB is there. Because by adding in water, you're removing TMB, all right? And so, and that's resulting in a result the same as the control. And if you go back here, when TMB is there, stuck on the seed, it stops germination from happening. But when you remove it, your results go all the way up to about the control group. So there's a lot of things that are happening here. So let me hopefully kind of clean this up a little bit. So when you're looking at these, let's kind of just analyze and make some predictions about what's going on with this experiment. So it's nothing that I think is going to be too terrible to write out, but a lot of this does involve just your analytics on how do you perceive the variables and what's actually happening here. So in terms of this, let's take a second and analyze the key points here. So first, let's look at CAR. CAR itself, KAR, when you added in water, okay, compared it with, let's look at groups 2 and group 5, right? Because that's when you're adding in CAR. They're both going to be different from the control. So basically that means that even in the presence of water, you can't necessarily get rid of CAR. So that means regardless water present or not, CAR is going to actually help increase that germination rate. So when you compare groups 2 and 5, okay, this is showing you that CAR is always going to increase the germination, right? Now, let's jump back to, and I'll just kind of write this off to the side. Let's change this color. Let's say when you compare 3 and 6, okay? Because this is when you looked at TMB. All right, TMB, you need to understand that water is going to remove TMB, which is going to increase germination when TMB is removed. Okay, so basically that was kind of what you had to put together and put that in some support in terms of a grammatically correct sentence. So make a claim the effect about rinsing on the binding of KAR. So let's go back here. Let's just kind of answer this. KAR, rinsing doesn't have any impact, okay? Because when you look at it, KAR, when you compare groups 2 and 5, water or no water, it's going to be essentially different than the control. It's the same. And that means that CAR doesn't get removed. So think of it, when you add it in water, this KAR still stayed stuck onto the seed. It's going to help increase that germination. So what about the rinsing and binding of TMB? That's kind of what we went over here. That TMB is polar. So when you added water, it got removed, and that's going to help increase the germination. So that is key right here. When you compared groups 2 and 5, that's talking about CAR. When you compared groups 3 and 6, you essentially got different results. So when you compare these 3 and 6, groups 3 and 6, adding water caused it to have different results, right? Meaning when you removed TMB, your germination went up. So Let me just show you the rubric on this one really quick. It is insane in terms of what they were showing you on this template. So let's kind of make our best explanation for each one of these, and I think it goes pretty easy. So if you look at this template, it says the claims that car remains bound after rinsing, and that's kind of what we talked about. That's when you compare groups 2 and 5, so you got your 2 points from that one. TMB doesn't remain bound when you compare groups 3 and 6, and you got different results. So that's kind of what you were putting in. That's what they were anticipating you guys explaining on that one. And make sure that in your responses you are talking about the difference of the groups, okay? So that is that on part, on part B. Now, let's wrap this one up. So for this one, the last kind of summary here, yeah, this is the same thing. For C, it's asking, there is intense competition by plants to successfully colonize areas that have been recently cleared by fire. Describe one advantage of CAR regulation and one advantage of TMB regulation to plants that live in ecosystems of regular fires, okay? So it's pretty standard based on what the data shows you. CAR, even when you look at this information as well, CAR is always going to be a benefit. CAR always is going to increase germination. So CAR, a simple thing that I would state is going to help increase germination. But let's talk about TMB and how does that actually serve as a benefit? Well, if you think about it, TMB stops germination. And why is that a good thing? Well, if you think to this experiment, when you added in water, adding in water removed TMB, and that means you get the results the same as the control. Well, if you go back to the control, remember your control group, that's pretty darn high, a pretty darn good germination rate. So TMB actually is going to basically stop germination unless water is available. So basically that means that this seed is going to know when there's enough water to actually germinate. So TMB, you might think, oh, that's not necessarily a good molecule to have on the seed. It actually is, because that means that in order to get it removed, you add water, you're removing that TMB, and then it lets the seed know that there's enough water to actually germinate. So that is kind of the benefit. So basically this is just stating the same thing. What's the advantage of CAR? There's a lot of things you actually could have stated for CAR. It occurs at an increased time. You could have said that it actually germinates more and compete with other plants. And it occurs when fewer competitors are present / land is barren. I would just state that CAR increases germination. That's a pretty broad, easy point to be had there. But what's the advantage of the TMB regulation? That's basically stating that it's only going to germinate when enough resources, really water, is available. So that is the 2017 FRQ number 2.